Homo Sapiens, who are we, where do we come from? In Chapter 18, we will continue our look at the beginnings of life on Earth. We will primarily be concentrating on the abiogenesis of life, that is to say, the emergence of living cells from non-living organic molecules. As we noted in Chapter 17, there is no standard model agreed upon by scientists as to how life emerged from non-living matter. There are a handful of hypotheses as to how abiogenesis may have occurred on our planet. We will explore some of these hypotheses and see if we can arrive at any understanding about the appearance of life on Earth. As in Chapter 17, we will again be dealing with various aspects of complex biochemistry. At the sake of oversimplification, I will attempt to avoid complex explanations when possible. Let's begin Chapter 18 with some basics. The fundamental unit of life is the cell. The sin ancestor, or the last common ancestor of all life on Earth, was most likely a single-celled organism. The central question of abiogenesis then lies in how did the first living cells come into being. Cells, or living organisms, have characteristics that separate them from non-living materials or objects. Some of the primary characteristics of living organisms would be: 1. They undergo metabolism, that is, they carry out chemical reactions that provide energy for vital processes as well as synthesizing new organic material. 2. They maintain homeostasis, that is, they regulate themselves against environmental variables to maintain internal stability so that vital metabolic processes can take place. For example, being warm-blooded is an aspect of homeostasis in mammals. 3. Living organisms react and respond to external stimuli through sensory receptors. This allows them to maintain homeostasis as well as to interface with their external environment. 4. Living organisms grow. They are born, they live, and eventually die. 5. Living organisms reproduce. Their offspring are genetically derivative of the parent. They evolve over time as their populations are culled by natural selection, mediated by environmental and competitive factors that serve as filters selective for favorable genetic mutations. These fundamental characteristics of living organisms at the most basic level are mediated by chemistry and physics. Electrons, protons, and neutrons form atoms. Atoms combine together to form molecules. Molecules combine into complex systems that can become self-replicating. Self-replicating molecular systems, through some process of abiogenesis, become entities or cells, thus becoming the first forms of life. The transformation of these self-replicating molecular systems into living cells brings us to the crux of abiogenesis. How does a self-replicating molecular system or synergistic group of systems become a living organism? Let's now take a look at the main hypotheses as to how a living cell emerged from a non-living molecular system. Back in February of 1871, Charles Darwin casually hypothesized on the original spark of life in a letter to Joseph Hooker. Darwin wrote that the beginnings of life may have occurred in a warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, and etc. present, that a protein compound was chemically formed, ready to undergo still more complex changes. This early musing of Darwin was further elaborated on by Alexander Oparin and John Haldane in the 1920s. Oparin and Haldane hypothesized that life began in a primordial soup of organic molecules in a reducing atmosphere, that is to say, an atmosphere essentially free of oxygen. This primordial soup hypothesis was succinctly summarized by Robert Shapiro as follows. 1. The atmosphere of the early Earth was a chemically reducing atmosphere rich in hydrogen, methane, and ammonia, and essentially free of oxygen. 2. This reducing atmosphere allowed the production of simple organic compounds or monomers in the presence of various forms of energy such as sunlight, electricity, and heat. 3. On this early Earth, organic compounds accumulated in a soup concentrated along shorelines or oceanic vents. 4. This soup provided a favorable environment for the transformation of the monomers into more complex organic polymers, which eventually led to the first forms of life. In late 1952, graduate student Stanley Miller and Professor Harold Urey of the University of Chicago conducted the famous Miller-Urey experiment, in which they tested the primordial soup hypothesis proposed by Oparin and Haldane. Miller and Urey designed an apparatus through which they cycled a mixture of water, methane, hydrogen, and ammonia in the presence of electrical sparks, this being a crude simulation of the hypothesized reducing atmospheric conditions on the early Earth. After 1 week of operation, Miller and Urey found that about 10 to 15% of the carbon in the experimental apparatus had been transformed into organic compounds, including amino acids, the building blocks of proteins. In 2011, modern analytic techniques were used to reanalyze Miller's original experimental vials from 1952. The new analysis revealed that over 40 amino acids and amines had been synthesized in the Miller-Urey experiment. While the results of the Miller-Urey experiment showed promise, there were some caveats. The experiment also produced substances that tended to act detrimentally on the formation of more complex organic molecules. Add to this that the current scientific consensus has the Earth's early atmosphere being only weakly reducing or possibly neutral. A weakly reducing or neutral atmosphere would also be detrimental to the amount and variety of amino acids that could be produced in a primordial soup. Now, the primordial soup hypothesis and the Miller-Urey experiment dealt with the synthesis of organic molecules on the surface of the Earth, but research has shown that organic molecules can also be synthesized off-planet in outer space. In 2009, NASA reported that the amino acid glycine, one of the building blocks of life, had been discovered in material collected from Comet Wilt 2 by NASA's Stardust mission. In 2012, astronomers at the Copenhagen University reported the detection of the sugar molecule glycolaldehyde in a protostellar binary system 400 light-years from Earth. Glycolaldehyde is needed to form RNA or ribonucleic acid, which mediates key functions in living organisms. A fragment of the Murchison meteorite, which fell over Australia in September of 1969, was analyzed in 2010 and found to contain 14,000 molecular compounds, including 70 amino acids, the building blocks of the proteins essential to life. This seems to point to the probability that organic molecules were already present in the collapsing molecular clouds that gave birth to our solar system and our own planet Earth. So, the primordial soup synthesis of organic molecules, along with the potential for the extraterrestrial sourcing of organic molecules, seems to indicate that the necessary building blocks for living organisms were readily available on the early Earth. But the musing of life's spark occurring in Darwin's warm little pond faced a serious challenge. That challenge was that the surface of the early Hadean Earth was a very hostile environment for the abiogenesis of life. The period of the late heavy bombardment, which occurred roughly around the 4 billion year mark in Earth's history, was hypothesized to have sterilized the Earth's surface down to tens of meters in depth. The Sun was also in its T-Tauri stage of evolution, producing high levels of ultraviolet radiation detrimental to the appearance of life on the surface of the Earth. So, if the surface of the Earth was hostile to early life, perhaps life found more favorable conditions deeper in the Earth's surface. Professor Everett Schoch, in his paper titled Geochemical Habitats in Hydrothermal Systems, wrote the following: a logical deduction from the 16S ribosomal RNA phylogenetic tree is that the setting for the emergence of life had little, if anything, to do with the atmosphere or surface of Earth. Professor Schoch proposed that life may actually have found its origin in hydrothermal ecosystems such as deep-sea thermal vents or other subterranean geothermal ecosystems. As we discussed in Chapter 17, the deepest branches of the phylogenetic tree of life on Earth are populated by hyperthermophiles. Hyperthermophiles are organisms that thrive in extremely hot aqueous environments where temperatures range from 60 to 122 degrees Celsius. This seems to point to the earliest forms of life possibly being hyperthermophiles themselves. There is also the possibility, as Professor Schock points out in his paper on geochemical habitats, that perhaps the earliest organisms rapidly invaded all possible habitats, but their metabolic strategies became impossible once the O2 levels rose after oxygenic photosynthesis appeared. If so, it may be that hydrothermal systems are the only habitats that still host deeply branching organisms because those habitats remain largely unchanged since early in Earth's history. If then hyperthermophiles are just a remnant of a much more diverse population of early organisms, then the exact nature of Earth's first life forms may be difficult to discern. Be that as it may, there are still basic attributes that the first biological systems had to exhibit in order to become living organisms. Let's take a look at the most basic aspects of what was needed for the first life forms to appear. One of the main challenges in moving from a prebiotic state to a biotic state is the daunting amount of complexity involved in even a simple single-celled organism. Besides the complexity challenge, we also face the hurdle of having a complex array of biochemical systems that must work in synergistic concert in order for the organism to thrive. A cursory list of necessary biological imperatives would include 1. A membrane of some form to separate and shelter the organism's biological systems from a hostile world. 2. The organism would require a metabolic system for furnishing its energy requirements and building organic material. 3. The organism would need an information handling system for storing and retrieving genetic information for reproduction, as well as information for carrying out vital cellular processes, essentially some form of DNA. As we've seen earlier, the synthesis of basic organic molecules essential to life seems to be a given in our neck of the universe, whether terrestrial or extraterrestrial in origin. So, the building blocks of life seem readily available. The crux of the issue becomes by what process or series of processes do these organic compounds combine and transition into a living organism. Let's take a look at the current hypotheses on the origin of the 3 biological requirements of life that we just covered, namely a membrane, a metabolic system, and an information handling system. A membrane or some form of boundary is necessary to separate and protect an organism's life processes from the external environment. It would seem logical to assume that some form of protocell or vesicle would be a fundamental step in the beginnings of life. A protocell is defined as a prebiotic, self-organizing, self-ordered collection of lipids, lipids being a group of naturally occurring molecules that includes fats, waxes, and sterols. To quote from a paper by Irene Chen and Peter Valda titled From Self-Assembled Vesicles to Protocells, in a quest to build a simple living system, much recent interest has focused on encapsulating a genetic or metabolic system inside membrane vesicles. Vesicles are supramolecular aggregates containing an aqueous interior that is separated from the bulk solution by one or more bilayers of amphiphiles. Amphiphiles, to digress for a second, are chemical compounds possessing both hydrophilic or water-loving properties and lipophilic or fat-loving properties. Phospholipids, for example, are a class of amphiphilic molecules that are the main components of biological membranes. Stated succinctly then, protocells are naturally occurring membrane-like vesicles capable of housing the metabolic and genetic systems necessary for life. They form naturally in watery or aqueous environments. Without delving too deeply into the complex electrochemical nature of the protocell, let's look at some basic protocellular properties which make the protocell a plausible stepping stone to the beginnings of biotic life. The prebiotic protocells existing in their aqueous or watery environment would have been subject to the forces of natural selection, just as living organisms are subject to natural selection. Protocell or vesicle evolution might have been driven by the encapsulated contents of the protocell. To quote again from Self-Assembled Vesicles to Protocells, Darwinian competition between cells could have emerged simply on encapsulation of replicases or metabolic systems inside vesicles. Empty vesicles and vesicles encapsulating less efficient systems would shrink by losing membrane. This competition has interesting implications for the natural selection of chemical traits in the genome and membrane. Because counter-ions cause most of the change in osmotic pressure after polymerization, protocells containing charged polymers as the genetic material, such as RNA or DNA, would outcompete those with neutral polymers, such as a peptide nucleic acid. Again, at the expense of oversimplification, the prebiotic protocells, which encapsulated molecules exhibiting advantageous physiochemical properties, were more stable. These stable protocells outcompeted those protocells which were less stable. To again quote Chen and Valda, thus the competition arising from the simple physical properties of an encapsulated replicase could result in a snowball effect on the evolution of cellular complexity. The protocell could then become a possible first stepping stone to the emergence of life. Let's next look at a hypothesis for the emergence of prebiotic metabolic systems and ideas for the emergence of life associated with that hypothesis. Metabolism is defined as a set of enzyme-catalyzed chemical reactions that allow an organism to thrive. The metabolic system of an organism provides energy for cellular processes as well as building cellular components such as proteins and nucleic acids. Research suggests that the chemistry of metabolism predates the origin of life. The iron-sulfur world hypothesis is an origin of life hypothesis proposed by Gunther Wasserschauser, postulating that a metabolic system arose first and was later encapsulated by a lipid membrane. Wasserschauser proposed that the earliest form of life, which he termed a pioneer organism, originated in an undersea volcanic hydrothermal flow at high pressure and high temperature. As a detailed explanation of the iron-sulfur world hypothesis involves some rather arcane jargon and daunting chemistry, I'm going to oversimplify my explanation of the hypothesis for the sake of brevity. Dissolved volcanic gases such as ammonia, hydrogen sulfide, and carbon monoxide pass up through the rocky mineralized chimneys of the undersea vents in a pressurized, high-temperature flow of water. These volcanic gases react chemically with catalytic transition metal solids such as iron sulfide and nickel sulfide, which are found in the walls of the volcanic vents. Chemical reactions occur on the surface of the iron and nickel sulfide particles, creating an autocatalytic surface metabolism that produces organic compounds. It is hypothesized that once this primitive autocatalytic metabolism is established, a cascading cycle of ever more complex organic compounds and metabolic pathways are produced. William Martin and Michael Russell elaborated on Wasserschauser's hypothesis with their own ideas on the origin of life in deep-sea hydrothermal vents. They hypothesized that microcaverns in the walls of the hydrothermal vents provided a means of concentrating the newly synthesized organic molecules, thereby increasing the chance of forming oligomers or macromolecules. The steep temperature gradient within the hydrothermal vent allowed for a range of optimal thermal environments in which a variety of chemical reactions ranging from monomer synthesis to oligomerization could take place. The flow of hydrothermal water through the volcanic vent provided a constant source of building blocks and energy for the emerging prebiotic systems. Through a series of cascading steps, cellular evolution could take place in a single nurturing structure, the volcanic vent. The process of evolving through prebiotic chemistry to monomer and oligomer synthesis, to peptide and protein synthesis, to an RNA world, to ribonucleoprotein assembly, to a DNA world could take place in a single structure, facilitating exchange between all developmental stages. In this scenario, the microcaverns in the volcanic vent wall act as the cellular membrane, protecting the evolving cellular systems, with the synthesis of a lipid membrane being the last step in forming a free-living organism. Thus, in the iron-sulfur world hypothesis, the cell membrane becomes the final step in the emergence of life, with metabolic and genomic systems appearing first. So to quickly sum up to this point, we have the chemistry surrounding naturally occurring vesicles or protocells and the chemistry of hydrothermal ecosystems capable of generating two of the three biological imperatives we listed earlier, namely the cell membrane and a metabolic system. Let's now take a look at the third biological imperative, an information handling system. That is, some form of early genomic entity that serves as a precursor to DNA. The third biological imperative that we listed earlier is an information handling system, that is to say, a form of DNA. Our first biological imperative, the cell membrane, creates a stable environment for housing and protecting the cellular biological systems. Our second biological imperative, the metabolic system, provides energy for cellular functions and helps produce the necessary organic compounds the cell needs to thrive. Our third biological imperative, the information handling system, stores genetic data as well as the recipes and necessary biochemical information that the cell needs to function and reproduce. This information handling system is essentially a data storage and retrieval system. In our world today, DNA, deoxyribonucleic acid, is at the core of the information handling system used by all life on Earth. The hypothesized precursor to our present DNA world is the RNA world, a world in which ribonucleic acid is the centerpiece of life. Before we dive into the details of the RNA world hypothesis, let's take a look at the RNA molecule as compared to the DNA molecule. The RNA molecule is a polymeric molecule, that is to say, a macromolecule composed of many repeating subunits. These repeating subunits in both the RNA and DNA molecule are called nucleotides. A nucleotide is composed of three basic parts, a phosphate group, a ribose sugar, and a nitrogenous base referred to as a nucleobase. Both RNA and DNA use four nucleobases to build the nucleotides that form their macromolecular structures. DNA uses the nucleobases adenine, cytosine, guanine, and thymine. RNA uses the nucleobases adenine, cytosine, guanine, but replaces the nucleobase thymine with the nucleobase uracil. RNA and DNA also differ in the structure of their sugar components. The ribose sugar component of the nucleotide in RNA is composed of 5 carbon atoms, 10 hydrogen atoms, and 5 oxygen atoms, hence the chemical formula C5H10O5. The sugar component of the nucleotide in DNA is composed of 5 carbon atoms, 10 hydrogen atoms, and 4 oxygen atoms, which gives us the chemical formula C5H10O4. The loss of the one oxygen atom at the 2' location in the DNA sugar component is the reason behind the derived deoxyribose name. And just to digress for a moment for some nucleotide trivia, if we look at the nucleotide of RNA or DNA, we would note that there are missing oxygen and hydrogen atoms from the ribose sugars when referenced against their chemical formulas. This discrepancy results from 2 oxygen atoms and 4 hydrogen atoms being lost to 2 condensation reactions when the ribose sugar is chemically bonded to the phosphate molecule in the nucleobase. Each condensation reaction produces 1 molecule of water or H2O, which accounts for our 2 missing oxygen atoms and 4 hydrogen atoms. Getting back on track, the occurrence of the deoxyribose sugar in DNA's molecular structure gives us the derivation of its name, deoxyribonucleic acid, with RNA being ribonucleic acid. If we take a quick look at the structure of the ribose and deoxyribose sugars, we can see that the RNA molecule has a hydroxyl group at the 2' position of the ribose sugar component. The DNA molecule lacks this hydroxyl group at the 2' position. A hydroxyl group being an oxygen atom connected to one hydrogen atom by a covalent bond. The presence of this extra hydroxyl group makes the RNA molecule less stable than the DNA molecule. This instability arises when the RNA molecule is not constrained in a double helix, as this can allow the 2' hydroxyl group to chemically attack the adjacent phosphodiester bond, cleaving the phosphodiester backbone of the RNA molecule. A phosphodiester bond in the case of RNA or DNA is the chemical bond that binds the phosphate group to the ribose sugar molecules of the nucleosides. The phosphodiester backbone refers to the sugar phosphate polymeric chain that binds and supports the nucleobases in the RNA and DNA macromolecule. RNA's use of uracil in place of the thymine found in DNA can also be an issue, as a nucleotide constructed with uracil can render RNA more susceptible to mutations. So, in general, the chemical properties of RNA make it a less reliable information storage system than the more robust DNA. But as a precursor to the current DNA standard, RNA was sufficient to the needs of the first prebiotic entities, with the added advantage that RNA was capable of playing crucial roles that DNA could not. Let's now take a look at the RNA world hypothesis. The RNA world hypothesis postulates that RNA, or ribonucleic acid molecules, were precursors to all current life on Earth. As evidence to this, we find the following clues. 1. Many of the most critical components of present-day cells are composed largely or entirely of RNA. 2. RNA is critical to the process of translating the biochemical recipes stored in DNA into proteins critical to the functioning of a cell, as evidenced in transfer RNA and messenger RNA. 3. Small RNAs can catalyze all of the chemical group and information transfers required for life. 4. The ribosome, the site of protein synthesis within the cell, has a central core composed of RNA. And as a precursor to DNA, RNA has the advantage of being able to perform both the data storage tasks of DNA as well as act as an enzyme. That is, RNA can catalyze chemical reactions. Because of RNA's ability to fulfill these dual roles, RNA is believed to have been once capable of supporting independent life forms. As promising as the RNA world hypothesis is, there are still some concerns that need to be addressed. One basic concern is how were some of the chemical constituents of RNA synthesized prebiotically. There are also concerns as to whether ribose and other backbone sugars that compose the RNA molecule are stable enough to have been found in the original genetic material of the first life forms. Some of these perplexing questions are being addressed by ongoing research that continues to break new ground in support of the RNA world hypothesis. As an example, in a research paper tackling the question of prebiotic synthesis of RNA constituents presented by Powner, Gerlaw, and Sutherland of the University of Manchester, titled Synthesis of Activated Pyrimidine Ribonucleotides in Prebiotically Plausible Conditions, the researchers state the following. At some stage in the origin of life, an informational polymer must have arisen by purely chemical means. According to one version of the RNA world hypothesis, this polymer was RNA, but attempts to provide experimental support for this have failed. Here we show that activated pyrimidine ribonucleotides can be formed in a short sequence that bypasses free ribose in the nucleobases and instead proceeds through arabinose aminooxazoline and anhydronucleoside intermediates. The starting materials for the synthesis, cyanamide, cyanoacetylene, glycolaldehyde, glyceraldehyde, and inorganic phosphate are plausible prebiotic feedstock molecules, and the conditions of the synthesis are consistent with potential early Earth geochemical models. Basically, what the researchers are saying is, through chemical processes and conditions hypothesized to have existed on the early Earth, the prebiotic synthesis of RNA pyrimidine ribonucleotides is possible. As we covered earlier, the RNA macromolecule is composed of repeating subunits called nucleotides. These nucleotides being composed of three basic units, the phosphate group, the ribose sugar, and the nucleobase. The RNA and DNA nucleobases fall into two groups, purines and pyrimidines. Adenine and guanine are purines, with thymine, cytosine, and uracil being pyrimidines. In the double helix bonding scheme of RNA and DNA, the purines bond with the pyrimidines along the core of the double helix. Adenine, the purine, bonds with the pyrimidine thymine in DNA in the pyrimidine uracil in RNA. Guanine, the purine, bonds with cytosine, the pyrimidine, in RNA and DNA. Now, if we take a look at this schematic depicting a series of chemical reactions as set forth by Pauner, Gerlaw, and Sutherland, we can get a basic idea of how a pyrimidine ribonucleotide, which is composed of a phosphate group, a ribose sugar, and a nucleobase, can be synthesized from prebiotic feedstock molecules. Starting with cyanamide and glycoaldehyde in an aqueous setting with phosphate present, we can get a chemical reaction that forms 2-amino-oxazole. The 2-amino-oxazole reacts with glyceraldehyde to produce arabinose aminoxazoline, glyceraldehyde being a monosaccharide or simple sugar. Arabinose is also a simple sugar similar to the ribose sugar that is a component of the nucleotides that form RNA, both arabinose and ribose being classified as aldopentose sugars. Aminoxazoline is a molecule composed of oxazoline, a heterocyclic or ring-structured 5-membered compound containing 1 atom of oxygen and 1 of nitrogen, along with 3 carbon atoms with an attached amine, the amine here being the nitrogen atom bonded with 2 hydrogen atoms. The arabinose aminoxazoline then becoming a cursory chemical form of a nucleoside. The arabinose aminoxazoline reacts with cyanoacetylene to produce an anhydroarabinose nucleoside. This arabinose nucleoside intermediate, in the presence of diphosphate, urea, and ammonium ions, undergoes phosphorylation, that is, bonding with a phosphate group. The phosphate group attaches at the '2' and 3' carbon locations of the sugar molecule of the synthesized pyrimidine nucleoside, with the end result being cyclic cytidine 2', 3' monophosphate. Further chemical transformations can then yield cytidine monophosphate, a nucleotide that is one of the building blocks of the RNA macromolecule. This promising research seems to answer some of the questions surrounding the prebiotic synthesis of pyrimidine ribonucleotides on the early Earth, though the chemical pathway to the purine ribonucleotides is still being sought. NASA has also done research on the organic materials found in meteorites that suggest that the building blocks of RNA, such as adenine and guanine, can form extraterrestrially in outer space. As stated earlier, astronomers at the University of Copenhagen detected the specific sugar, glycoaldehyde, 400 light-years away in a distant star system. Glycoaldehyde plays a key role in the synthesis of RNA nucleotides, as we saw earlier in the research on the prebiotic synthesis of pyrimidine nucleotides. The work of NASA and other researchers points to the possibility that complex organic molecules can form in stellar systems prior to the formation of planets, with these organic molecules eventually becoming incorporated into the fabric of the planets themselves. Through geochemical processes, as we discussed earlier, these organic molecules can then be transformed into more complex organic compounds that become the wellspring for the appearance of life. Given basic geochemical processes along with a source of fundamental organic molecules, it seems possible that life may be able to arise wherever conditions are ripe. Whether the RNA world hypothesis proves to be the correct model for the emergence of life or some other model arises, there seems to be little doubt that RNA played a key transitional role in moving life forward to the DNA world we know today. So, to quickly summarize what we have explored so far, we've looked at ways and means by which abiogenesis can 1. Generate a cell membrane through naturally occurring processes and conditions hypothesized to have existed on the early Earth. 2. Give rise to a metabolic system via chemical reactions occurring in a nurturing, geologically active hydrothermal ecosystem. 3. An information handling system, most likely RNA or some earlier precursor, can be generated through natural geochemical processes and conditions such as those that were hypothesized to have existed early in the history of the Earth. Therefore, from a scientific standpoint, the hypothesized fundamental prebiotic processes at the crux of abiogenesis appear to be viable. The missing step that has escaped researchers so far is how these prebiotic systems made that final leap to becoming a living organism, Darwin's spark of life. In Chapter 19, we will continue our look into abiogenesis with a journey along the shadowy border that separates the prebiotic world from the biotic world in search of Darwin's spark of life.